Hi everyone, my name is Cheyenne Beach, and today I'm going to be talking about some main points from my master's research, which was evaluating selected physiological responses to sublethal Cyathocotyle bushiensis and Sphaeridiotrema species experimental infections in captive lesser Scaup. Now I know that sounds like a lot, but we're going to work through it together. So first, I'm going to give you guys some background information, get you up to speed on the issue at hand. Then I'm going to answer the question, what does the physiological response to trematode infections look like in Scaup? And then lastly, wrap it all up, talk about some management implications. So lesser Scaup, they are a member of the diving duck guild. This means that they are physiologically adapt to foraging underwater. They are the most abundant diving duck in North America. They utilize almost all of North America for their breeding, migratory, and winter areas. In the United States, almost half of Scaup harvest occurs in the Mississippi flyway. So North America is split into 4 flyways. And for almost half of Scaup harvest to occur in one of those flyways means that it's pretty important in that area. And that area is the Mississippi River system. That being said, they're experiencing population declines and have been since the mid-1970s, without a clear indication as to why they're experiencing population declines or why there haven't been any rebound in population numbers like there have been for other waterfowl species. Reasons attributed to the decline include human development and agricultural development and the loss of wetland area and degradation of those wetland areas. The wetlands that are left on the landscape are at an increased risk of pollution. It's been found that Scaup prey species and diet items are in decline. And there's been increased competition found between Scaup and other wetland species such as fish. So all of these things compound off from each other. And they also don't really tell the full story. It doesn't clearly indicate why there's been a decline, and more importantly, why there hasn't been a rebound in population numbers. That's led some people to look into the role that episodic diseases play in population dynamics of Scaup. Particularly, we're talking about trematodiasis. That's the mortality and morbidity caused by trematode infections. These are facilitated by the exotic Faucet snail, and these trematode species are exotic. Faucet snail is the intermediate host to these trematode species, and waterfowl serve as the final host. Pictured here are what the trematodes look like. On the left, we have Cyathocotyle bushiensis, and on the right, we have a Sphaeridiotrema species. On the top is the metacercariae stage. This is the infectious stage to waterfowl. And on the bottom, we have what the adult trematodes look like in the intestinal tract of waterfowl. Most trematodiasis events occur in pools 7 and 8 of the Mississippi River. This is Wisconsin and Minnesota areas. And faucet snail distribution currently has reached as far south as pool 13 of the Mississippi River, which is just within the Illinois border. And managers are concerned as to what will happen to Scaup populations once the faucet snail and trematodiasis reach pool 19 of the Mississippi River, because this is a major stopover location for Scaup during migration. So that gets you guys up to speed and lets you know why I'm here today looking at this kind of stuff. That leads into the question of what does the physiological response to trematode infections look like in Scaup? To answer this question, we needed to get our hands on some birds. And we had a subset of birds that were wild caught. So these are wild birds captured and brought into captivity. We captured them in backwater areas of the Illinois River system and brought them to Forbes Biological Station and did the experimental infections at Forbes Biological Station. These birds were as close of a representative to a wild bird as we could get. They have experienced migration before. They, again, are completely wild. They have experienced parasitic infection before, and they may have come in contact with these trematodes before. Some drawbacks, however, is that they are not adept to living in captivity, and they have a great aversion to humans. So to offset or counteract the drawbacks of our wild-caught birds, we had a subset of captive reared birds. So we collected wild eggs from wild Scaup nests and brought those eggs to Forbes Biological Station to incubate and hatch in captivity. And we reared ducklings to, they were fully fledged. This was about 5 months in age. And these birds served as our blank slate. They've never experienced a parasitic infection before. They've certainly never come in contact to these trematodes before. And they have only lived in captivity before. So they are adapt to that environment, and they don't have as much of an aversion to humans as their wild-caught counterpart. So between the two captive status groups, we hope to fill some gaps that each may have. During the experimental infection trials, we collected a suite of morphometric measurements like mass and wing chord. We also collected blood samples at various benchmarks throughout the trials so that we could evaluate various metabolic functions and immune functions. For each captive status group, a subset were the treatment group. We also had a control group that were uninfected. The treatment group received an infectious dose, and we had a wild-caught above here. So that's the breakdown of their infectious dose, and our captive reared down here, that's the breakdown of their infectious dose. We tried to get these dosages as close as possible to each other as we could. However, there is natural variation, and so they're not perfect. I also want to point out that while each captive status group was under the same conditions during their trials, the trials did not happen at the same time. The trials lasted for 10 days, and on day 10, the birds were euthanized, necropsied. We kept the intestinal tract to evaluate the presence and abundance of trematodes, and we sent the carcass to the University of Arkansas Monticello for a full breakdown of the protein and fat content. Generally, our captive reared birds had less trematode abundance than their wild-caught counterpart, and our male birds had less trematode abundance than their female counterpart. Those are important little nuggets to hang on to. To get at this question of what's the physiological response look like, I selected just a small handful of the response variables that I looked into during these experiments. So this is just a small subset, but it explains the response well. So eosinophils, these are white blood cells that are specialized for parasitic infections. And these white blood cells are a part of the innate immune response. This is what vertebrates are born with, rather than an acquired immune response that you build after an infection. So you build up antibodies after an infection. We see that regardless of captive status, throughout time, eosinophil levels increase. And we also see that birds that had greater total trematode abundance had greater eosinophil levels. Selenium, this is an essential nutrient in the body. It's used for all sorts of things. One of the things is to help immune function. It helps buffer tissues from damage during an immune response, and it also helps the immune system function correctly. We see that birds that had greater total trematode abundance had lower selenium levels after infection, and to the point that it's below a minimum threshold. So we have a depletion of selenium. And body fat, just generally, wild-caught birds had lower body fat levels than their captive-reared counterparts, and this was true for our control birds and our treatment group. So just generally, they had lower fat levels. And female birds had lower fat levels than male birds. And again, that's generally across control groups and treatment groups. But what's really important is that when we look at infection intensity, trematode abundance. So in particular, Sphaeridiotrema species, infection intensity, so the abundance of Sphaeridiotrema in a body. Birds that had greater Sphaeridiotrema infection intensity had lower fat levels. So there's a depletion again of reserves. So what's happening to Scaup after a trematode infection? Essentially what happens is these birds, they get to an area that trematodes are occupying, and they get a lethal or sublethal dose, however much they happen to eat, and in the body an immune response is initiated. Again, this requires some selenium to help buffer tissues and to help the immune system function correctly. As the trematodes develop, we have increased tissue damage because they need to take in resources. This damage is happening to occur in areas where selenium uptake happens in the intestinal tract. So selenium input is decreased because of that damage. And also, we're not eating as much because we're not feeling as good. So selenium input decreased, but selenium output is still increasing because we still have an immune response and we still have damage occurring. This is coming to a breaking point where immune function is getting compromised. This either leads directly to mortality from direct impacts of the infection, or we're at a compromised state where immune function is decreased because we're tapping into all of these reserves and resources that we have in the body, and potentially a reproductive success is decreased because we're spending all of this time and energy trying to repair damage from the intestinal infections, and we need to rebuild resources. And we were essentially there to refuel and rebuild resources during migration. So we have this very energetically demanding period of time, and we need to build up resources for reproduction, but we're seeing a decrease in resources because of these infections. So we're left in a compromised state. So what can we do about this? Managers need to look for ways to mitigate the interactions between Scaup and Faucet snails and thus decrease trematodiasis events. We need to increase the quantity and quality of migratory stopover locations, especially in areas where trematodes do not occupy and trematodiasis doesn't occur. And we need to look into this further. We need to look into how reproductive efforts are impacted. We need to see if immunity is possible, if there are possible ways to get acquired immunity and build those antibodies, or if that is going to further lead to an overactive immune response. And we need to look into how long does the trematode life cycle need to be disrupted to decrease trematode populations. And this is directly impacting our mitigation efforts. So with that, I'd like to thank everyone involved in this project. Funding comes from the Federal Aid in Wildlife Restoration Act, as well as some funding from Ducks Unlimited Canada Institute for Wetland and Waterfowl Research. I have a fellowship with them. Conducted my research at Western Illinois University, and I had a whole slew of people that helped out. University of Arkansas Monticello, University of Illinois Urbana-Champaign, the Illinois Natural History Survey, U.S. Geological Survey, and folks at Forbes Biological Station. Thank you. Are there any questions?